Welcome to this series of preclinical sciences for medical students. Our unit of discussion is Biochemistry of Hemoglobin, and our lesson is on Pophyrins chemistry. Taking you through this lesson is I, Dr. Joroge, a practicing clinical biochemist, toxicologist, and an expert in phytotherapeutics pharmacognosy. On completion of this lesson, you will be able to describe the pophyrins, illustrate the structure of pophyrins and their isomers. You'll be able to classify the pophyrins, describe the properties of pophyrins, discuss their biological importance and applications. What are pophyrins? Pophyrins are large organic compounds composed of continuously connecting heterocyclic atoms. They are ubiquitous in nature and present in mammals, fish, birds, petroleum, plants, and microorganisms. They are naturally occurring and can be synthetically produced. The pophyrins are made up of basic units known as the pyrols. The pyrols are heterocyclic atoms made up of 4 carbon atoms, 5 hydrogen atoms, and a nitrogen atom. The pyrols have a molecular formula C4H5N. The nitrogen atom of the pyrrole is oriented at the center. Fusion of 4 pyrrole rings labeled 1, 2, 3, and 4 makes up a tetrapyrrole. This is the basic structure of pophyrins. The 4 pyrrole rings are linked together by methynyl or methane bridges. The tetrapyrrole has a molecular formula C20, H14, and 4. The pophyrins have a total of 26 pi electrons. The 18 pi electrons are conjugated to form a planar. Equal distribution of electrons allowed the tetrapyrrole ring confer stability and aromaticity of the molecule. According to Fisher's nomenclature, methynyl bridges are labeled as alpha, beta, gamma, and delta, and their positions are referred to as meso position. The alpha pyrulic substituents, carbon positions which are not attached to methynyl bridges, are numbered 1 to 8, and each pyrrole has 2. The side chains may be substituted by acetyl, propionyl, methyl, or vinyl groups. Combination of the substituted side chains results to development of a wide variety of pophyrins or isomers. Pophyrins having 2 different side chains will have 4 isomers, named in Roman 1, 2, 3, and 4. Examples of such pophyrins include etiopophyrin, uropophyrin, and copropophyrin. Pophyrins that vary in their side chains with 3 different substitution molecules form 15 isomers. Examples include mesopophyrin and protopophyrin. Protopophyrin IX is the last common precursor on the biosynthetic pathway of hemoglobin and chlorophyll. The pophyrins exist in 2 groups, and they are classified based on the arrangements of the side chains. In the type 1 pophyrins, the side chains are symmetrically arranged. Examples include uropophyrin I or 1. In type 3 pophyrins, the side chains are asymmetrically arranged. They have a reverse position of the methyl substituents group that occurs on the 4th ring. The type 3 pophylins are the most abundant in nature than type 1 and are more common in biological systems. Examples include uropophylins, copropophylins, and protopophylins. Heme, a type 3 pophylin, has the order of the substituent groups as MV, MV, MP, and PM. Forming the type 3 pophylins are the metallopophylins. The metallopophylins are pophylin metal complexes which are naturally formed when metal ions bind with the nitrogen atoms of the pyrrole links. A major example is heme, which has got a metal atom and is found in myoglobin, hemoglobin, certain cytochromes, and chlorophyll. Hemoglobin molecule is an iron-containing pophylin, while chlorophyll is a magnesium-containing pophylin. Shown here is a structure of a metallopophylin with the iron molecule or iron centrally placed. On the properties of pophylins, we look at aromaticity. The pophylins are very stable, that is, do not break so easily, and they have a wide range of reaction with other substances. This is largely attributed to the conjugated pi electrons. On solubility, the pophylins are highly hydrophobic and has low solubility for water. Their solubility in organic solvents depend on the functional substitution of the hydrogen atom in the pyrroles. This is highly significant in the administration of pophylins into biological systems. On their photophysic properties, the pophylins are deeply colored. They absorb strongly in the visible region of the electromagnetic spectrum. The Greek word porphyra, which means purple, is in reference or coined the term pophylin. The absorption spectrum of the pophylins depends on the structure and presence or absence of a metal. The pophylins transmit through an intense band between 390 and 430 nanometers, that is near ultraviolet. These are called the solid of the B band. They also transmit through a lower intensity band at longer wavelengths between 500 and 680 nanometers called the Q band. On fluorescence spectroscopy, the pophylins are characterized by two emission zones corresponding to energy transitions between 400 and 800 nanometer. The pophylins have got numerous biological functions. The primary function of the pophylins is the synthesis of heme. Heme is complexed with proteins to form heme proteins. It serves as the prosthetic group in hemoglobin, myoglobin, and the cytochromes. The heme proteins are integral components of essentially every cell of almost all living organisms, where they have diverse roles in vital life processes. The table illustrates the various functions of pophylins. Primarily, the transport of oxygen in blood that occurs due to hemoglobin in erythrocytes, the storage of the oxygen in muscle of animals, which is in reference to myoglobin in muscles, It's also involved in the electron transport in the respiratory chains in animals, plants, bacteria, yeast, and archaea. This is in reference to cytochrome in respiratory chain A, B, C, D, F, and O. The pophyllenes are also involved in the synthesis, modification, and or degradation of fatty acids, steroid and adrenal hormones, anesthetics, polycyclic aromatic hydrocarbons, and other xenobiotics. This is in reference to the cytochrome P450. In addition, the pophyllenes are involved in the activation and metabolism of hydrogen peroxide. This is in relation to the catalase and the peroxidase enzymes. The pophyllenes are also key in the breakdown of heme, where heme oxidase is largely involved. The pophyllenes are also used in the oxidation of tryptophan, where tryptophan pyrrolase is key, used in the insertion of oxygen atoms in organic substrates, where oxidases are applied. They are also involved in the synthesis and transport of nitric oxide that is involved in smooth muscle relaxation, platelet inhibition, immune regulation, neurotransmission, and penile election. This is in reference to nitric oxide synthase or NOS. In addition, the pophyllenes are involved in the metabolism of nitrogen and sulfur oxides, which is in reference to sulfite and nitrate reductase or sulfite oxidase. Pophyllenes are also key in oxygen sensing, binding, and release, mostly used in oxygen sensing proteins, the hemoprotein kinases, and direct oxygen sensor heme protein domain. In invertebrates and tissues, the pophyllenes are key as erythrochroins. The magnesium pophyllenes are mostly found in plants and are related or involved in photosynthesis. The breakdown products of heme are harmful to infants and alcoholics. The intermediates in the synthesis of heme causes the genetic disease called the porphyrias. Excessive heme breakdown of pophyllenes lead to the hyperbilirubinemia, condition that is accumulation of bilirubin in the blood and tissue, which is seen in neonatal jaundice of the infants or in genetic diseases such as Crigler-Najjar syndrome. Further, genetically modified forms of hemoglobin are responsible for sickle cell anemia and a number of methemoglobinemia. Pophyllenes have got numerous applications, and in medicine, they are used as antimicrobial and antiviral substances. For instance, the prevention of sequestration of heme to hemozoin, that is, preventing the formation of hemozoin by the malaria-causing organism Plasmodium falciparum, is used as a target for treatment of malaria using antimalarial drugs artemisinin, chloroquine, cinchona alkaloids such as quinine and quinidine, and various isonitriles derived from marine organisms. The pophyllenes are also used in cancer treatment and diagnosis. Porphyric pigments and their metal complexes localize with high affinity in neoplastic tissues than normal tissues. These characteristic is integral in photodynamic therapy of cancer. In phototherapy, the tumor is exposed to an argon laser which excites the porphyrins that emit cytotoxic effects or reactive oxygen species on tumor cells. Synthetically prepared or naturally occurring heme are investigated with great interest as diagnostic agents. We have come to the end of our lesson on porphyrins chemistry. Our next lesson will be on the biosynthesis of porphyrins. Welcome again, and thank you.